Okay, so I want you guys to imagine you're diving, scuba diving. This is what it sounds like when you're underwater, when you're breathing, using the respirator, and you're snorkeling around, looking at all the different marine life. This is some of the stuff that you're going to be seeing. Oh, stingray, all sorts of fish, corals, sponges, protists, etc. So huge diversity in the marine world. We talked about phylum Porifera in the last lecture. What we're going to talk about now is the phylum Cnidarian, or Cnidaria. So Cnidaria are completely going to be, well, I shouldn't say completely, the vast majority, about 99% of the Cnidaria are going to be marine species. There are a handful of freshwater species, and I'll show you some examples of these guys as we get into the lecture. But what I want to do is make sure you're comfortable with where do Cnidaria or the Cnidarians go in the classification scheme. So we're talking about, in the last lecture, we were talking about Porifera, and we talked about Parazoa. Let me get the little marker, little pen here. Okay, Parazoa is right over here. Those are animals that lack true tissues. They lack any type of body symmetry. The good news is everybody else that we talk about, all the other species, are going to be under this heading. Again, it's not a kingdom or a phylum, it's a general heading. So Eumetazoa is that grouping all the other animals we talk about go into. Now, what makes Eumetazoa the characteristic? I'm going to put this in a little text box. Make sure you guys are... anybody in Eumetazoa, there it is. AZOA means they have true tissues. So all the animals we continue to talk about from now on out go under Eumetazoa, and they have true tissues. So even though they're simple, these Cnidarians are pretty, for the most part, we consider them pretty simple animals. They do have distinct or true tissues in their body. So as I mentioned just a minute ago, these are by and large marine species living in the ocean, saltwater. The freshwater species that we'll talk a little bit about is called the hydra. We know of over 10,000 species, and this is a number that is continuously evolving because we keep finding more. So easy ones to remember or think about are, well, the hydra, jellyfish. Jellyfish fall under Cnidarians. Corals fall under Cnidarians. Those weird colonial animals that have those skeletons made of calcium carbonate. And then also another group called anemones. So if you saw Finding Nemo, Nemo lived in the anemone. The anemone would belong in the phylum Cnidaria, or the Cnidarians. Okay, so when we're looking at all of the Cnidarians, the big defining feature is the structure known as a cnidocyte. So you go across the Cnidarians, the cnidocyte is going to be present. This is, make sure I spell right. This is a stinging cell. So I want you to think about it as a gun. The stinging cell, or the gun, can shoot out little harpoons, little bullets, little things that don't feel so good, called nematocysts. The nematocyst, and I'll cover this more in a few minutes, is the thing that gets discharged. All right, so that is the key defining feature of Cnidaria, are the presence of cnidocytes. Now, what they shoot out, well, that varies. There's 20 different varieties that have been identified, but they all have these little stinging guns, which is why we don't we don't really want to play with them too much. Some of them are fun. You can touch them and it's not going to hurt you because of the type of stinging cell and the type of nematocyst discharged. Others, don't mess with them. They hurt bad. So, all right. So, looking across the cnidarians, we will see two body plans. The body plan rotates in some groups back and forth based upon which portion of the life cycle that they are using. So body plan wise, what we'll see is in large or in general, they are a sac with a central digestive cavity. This is known as a gastrovascular cavity. All right, so, or make sure I put up this abbreviation, GVC. That is the abbreviation for gastrovascular cavity. All right, so basically a bag. Now, take your bag and or your sack and stick it down on the ground and attach it to the ground, and you have what we call the polyp body plan. This one here. Okay, here's your sac. You got an opening on the top. That's the mouth and anus. Becomes the same structure. Food goes in, waste product comes out. That's real pleasant to observe. And there's your big sac. That's the digestive cavity. So polyps tend to be very sessile. They don't move around. They're not active movers for the most part. The hydra can do a little cartwheeling thing, but in general, polyps are sessile. So coral polyps, they don't move. Anemones tend not to move a whole lot. The other body plan is known as the Medusa. So if you guys are familiar with Greek mythology, Medusa was that crazy lady with the snakes in her hair. Well, take your sac and flip it upside down. And now it floats around like an umbrella. Looks kind of like an umbrella with tentacles hanging from it. Same sac. Here's your mouth, anus. Food goes in. Waste products come out. The yellow outlines the gastrovascular cavity. And then the sac just hangs upside down. And the tentacles hang downward. Now these guys are a little more mobile. Not real active. They're not going to chase you down. If you get stung by jellyfish, it's usually because you swam into them or the current pushed them into you. They're not swimming after you. Very minimal movement in these things. There can be a little, but not much movement at all. All right. Tentacles hanging down. The tentacles are where all of the cnidocytes will be found. That's where they're going to, those are the specialized cells that shoot the bullets. Those are found in the tentacles. Now, when we look at the general body plan as well, we look at the tissue groups that make it up. You have the outer tissue, which is the epidermis. You have this layer beneath the epidermis. That's called the mesoglia. It's kind of a jelly-like fluid, gives it a little bit of structural rigidity. I mean, they're not hard at all. They're soft and squishy, but that's kind of, again, that jelly-like consistency or jello-like consistency where it has a little bit of structure to it. And then the next layer of tissue will be the gastrodermis. It's going to be that inner layer of tissue. So notice they don't have the endodermis layer. They have an epidermis and a gastrodermis, and that's it. The gastrodermis makes up the inner layer there. That's basically their internal layer. And what we don't see on here, but what is present, I'm going to change font to, let me go to green, is that these guys have a very simple thing called a nerve net. So it looks like, well, a net that will be embedded within their tissues and runs up into the tentacles. All these little nerve fibers. In some of the jellyfish, it's kind of concentrates into a very, very primitive brain. That's kind of a stretch to call it a brain. brain-like structure, might be a better description, but the nerve net allows them to react to stimulus. So you touch one tentacle over here, there's our little fish, I'll give him a little tail, there's a little fin there, back fin, and that tentacle hits it, or it hits the tentacle, the nematocyst will discharge, the nerve net says, hey, something just swam into us, and then it can get the other tentacles to move over and grab it, discharge their nematocysts, and consume that organism, whether it's a little fish or it's plankton or whatever. So very, very basic neural tissue here. True tissue. This is why these guys go in eumetazoa and they're not sitting over there in parazoa. So because they have the true tissues. So, okay, so let's see. So terminology and structures to get very, very comfortable with. Cnidocyte. Cnidocyte is the gun. Nematocyst is the bullet. Bullets. And over 20, as I mentioned before, over 20 different ones have been recognized that can discharge. So this is recognized, the discharge of the nematocyst, shooting of that gun, is recognized as the fastest cellular process in nature. This is faster than a nerve impulse. So it's amazing when these things go, they go shooting out. So what will happen is all of these nematocysts or cnidarians have a cnidocyte, and they have a little trigger here. So that's our little trigger. It's like a gun trigger. When that trigger is touched, it sends the stimulus down, and that causes the nematocyst to discharge. So you can see over here the nematocyst has been discharged. This one's a sticky thread. So it discharges and this little thread coils around whatever prey has touched it. Other nematocysts come out and they look like little harpoons where they will actually spear into the prey. Others, different shapes for their harpoons, different types of bullets. One cnidarian we'll talk about in another lecture actually discharges its little harpoons and they are not still attached. They get shot out into the water column, floating away from the cnidarian, which is not good at all. And I'll go through a little story about those guys in a bit here. But a lot of different varieties here. But the key is the animal does not need to be alive to discharge this nematocyst. So you see a dead jellyfish washed up on the beach, you go and you grab it, you're going to get stung. So always word of caution, be careful if you're playing around with jellyfish or any of the cnidarians. The nematocysts they discharge can often contain a lot of toxins. They can be very uncomfortable depending upon the type of toxin. The degree of discomfort will vary. Now they're protein-based structures. So if you get this discharged into your body, into your skin, and it penetrates through your skin, you need to denature the protein to remove the discomfort. Okay, so this is where you get the episode of Friends, if you guys saw those reruns, that, oh, you got stung by a jellyfish, let me pee on you. So I want you guys to think about this. Your biology majors, you've talked about it, you've covered this. It should be something that you should remember for all of your future courses. There are two variables to denature a protein. Hopefully you guys just, those popped up in your head. And you went, oh, it's blank and blank. What two variables denature a protein? You change the shape of the protein doesn't function. So the protein hurts, change its shape. Would urine change the shape of those proteins? Does it meet those variables?